In the last lecture, we have discussed about the mechanism of urine concentration and dilution. So in that, we have answered two questions: why kidney make sometime concentrated urine, and why kidney makes sometime diluted urine. So in that, we have learned about how the concentrated urine is made by kidney and how the diluted urine is made by kidney. So we have learned by two examples that how the osmolarity of the body fluid is maintained. So now today we are going to discuss about the answer the third question, which was so how this osmolarity is maintained by the kidney. So here in the last lecture we have seen about the position of nephron in the kidney. So here in this diagram, you can see the nephron location in the different area of the kidneys. So first area is the renal cortex. Another area is the outer medullary cortex. Third area is the inner medullary cortex. And the last one is the papillary cortex. It is called as the corticopapillary region. So here we have seen how the fluid is filtered in the Bowman's capsule, and what is the osmolarity of that fluid when it comes in the tube. So this filtered fluid is called as the tubular fluid, and in this first region of the nephron, that is the in this diagram you can see so in the first region, that is from Bowman's capsule to the thin ascending loop of Henle. So here the osmolarity of the fluid, it remains same. So last time we have seen one example, the osmolarity of the body fluid is near about 290 milliosmoles, and we have considered it as a 300 for our understanding. So as you move from the cortex to the papillary region, there is progressive increase in the hyperosmolarity of the fluid, or in the simple way, you can say it at healthy kidney has hyperosmolar medullary interstitium. So always in interstitium, so there is a in healthy kidney, so it is having the hypoosmolar medullary interstitium. So why we call this as a hyperosmolar? So in your body, any fluid has osmolarity more than 300, it is said to be hyperosmolar. When our body fluid has osmolarity less than 300, so it is called as the hypoosmolar compared to the standard plasma osmolarity. So standard plasma osmolarity is how much? It is 290 milliosmoles per liters. So this also applies to the urine. If you are producing the urine more than 300 osmolarity, then it is said to be hyperosmolar urine. And opposite to that, so when you produce the osmolarity of the urine is less than 300, then it is said to be hypoosmolar urine. So for this mechanism, so for urine concentration and dilution, So you will need three things. One is hyperosmolar renal medullary interstitium or cortical papillary gradient. So how this medullary interstitium osmolarity is produced? Number two, counter current multiplier 1. Number three is urea recycling in the renal medulla. And first we will see about the how medullary interstitium hyperosmolarity is maintained and what is the role of vasa recta, which vasa recta plays a very important role in counter current exchange. So counter current mechanism or the counter current multiplier here is different and counter current exchange is different. So we will see one by one. First, we have seen how hyperosmolarity is maintained. Number two, how it is produced. We have seen. So at the first distal cannulated tubule. So there you can see that how proximal tubule. So how this second thin part is permeable to water and how it loses the solute. So sorry, loses the water and how that tubular fluid becomes hyperosmolar. So here you can see, suppose this is different arteriole and if efferent arteriole and efferent arteriole. So here primarily the plasma osmolarity is 300 when it get into filter. So here is the osmolarity becomes 300 because two-thirds substances are reabsorbed. That is 65% of sodium, 65% of water, 100% of glucose, and 100% of amino acid, biocarbs. So here in this first proximal tubule region, so all these solutes are, that is two-thirds substances are reabsorbed. Then suppose the fluid is coming from here, it is also 300, here it is also 300, here it is also 300. So why you say at this region, the osmolarity it remains 300. So reabsorption of the solutes and solvents are in same proportion and in the same ratio. In medical terminology, when such situation occurs, it is said to be the reabsorption of the substances and solutes and solvent is isomotically. So here solutes and solvent reabsorb isoosmotically. So here in this second statement we can see nephron, the initial osmolarity of the tubular fluid is 300, but in ascending loop of Henle, cells are working in different fashion. So from here, from the proximal tubule to the ascending loop of Henle, so here it is 300, here it is gaining the osmolarity. So this part, the thin ascending part of loop of Henle, so it is water permeable and water loses from here into the interstitium and the osmolarity it increases. So I have explained you. So this region has to maintain the difference of 200 comparing to the interstitial fluid. Suppose here it is 200, the interstitial fluid should be 400. So here's, for example, here it is suppose 300. It is here losing the water and it becomes 400. So 400, 400 and this reaches 100 up to this. But when the descending, when the nephron, thin loop of Henle, it goes into descending position. So here first the descending thin part of loop of Henle, then descending thick part loop of Henle, then distal cannulated tubule, and the collecting tubule and late distal collecting tubules. So here first we will see so what happens in the thick region to the fluid. So here in the thick region of descending cannulated tubules, there are cells known as the principal cells. So how these principal cells, these cells, this ascending loop of Henle, these cells works in the different or in a special way. So first in this, try to understand this diagram. So I have drawn here only one cell. So here in this cell, this is the lumen of the descending thick part of loop of Henle. And here on this lumen, so here suppose this one cell is, I have drawn one cell, there are 100 of cells. So here this I have drawn one cell and how this cell used to function or the work. So in the lumen of this thick loop of Henle descending, the special transporters, types of transporters are present on the luminal side. So not on the basolateral side of the cell. So in this luminal side, so there are some special type of transporter proteins are present. And here in this thick part of ascending loop of Henle, so this special type of transporters have sodium, potassium, and 2-chloride here, and it makes the channels or the transports sodium, potassium, and 2-chloride inside the cell, right? So here in this part, sodium, potassium chlorides are transports outside to the interstitium. This part is called as the dilution part. So here in this principal cell, there are special type of proteins are present in the basolateral membrane. These proteins are nothing but they are the sodium, potassium, and chloride channels. So whatever the from the tubular fluid, so when this fluid reaches here, the concentrated fluid, suppose initially it is 300, then after it loses water and it comes this fluid that is for 400 up to the ascending loop of Henle, thick part. Then this fluid, it founds that there are some proteins, some transporter proteins are present in the luminal side. So then this protein, whatever there are the ionic concentration into this solute, which is tubular fluid. So this will take out the sodium, potassium, and 2-chlorides into principal cell. And this principal cell is having some special type of proteins. And these proteins are nothing but they are the sodium potassium channels. So these sodium potassium channels, so it will transport two chloride, sodium, and potassium out of these cells into the interstitium. So first the fluid is coming, so it is 300. So here it founds that is it is increasing 400. Again it will lose the water, and again it will dilute the interstitial fluid, and the concentrated fluid is coming here. So now we are going to the next part. So we are talking about the proximal tubule cells. So when these cells work to its highest capacity, can generate gradient difference between the interstitial fluid and interstitium. So generate difference of 200 with its maximum capacity. So here you can see the primarily the 300 is coming here. So when it comes into the thin part of descending loop of Henley, so after losing the water, so here 600, so here it becomes 400. So right, when fluid is going down and down, more losing more water. So here fluid, this 600 is coming here and it goes here. And here are the some proteins, transporter proteins. So from this fluid, it will take sodium, potassium, and the 2 chloride into the cell. And from the cell, the potassium is loose, it goes to the interstitium, it increases the hyperosmolarity. So here chloride ion also contribute, potassium ion also contribute. So this cycle keep on repeating, providing more and more hyperosmolar fluid. So here in this way, the current of the fluid is moving in counter direction. So here the current was 300, then it becomes 400. Again, this cycle is repeating. So this mechanism is called as the counter current mechanism. So descending segment provides hyperosmolar fluid. Until hyperosmolar fluid, it becomes 1200 milliosmol per liter. So when the fluid is coming from the filter position Bowman's capsule, so when it comes at the proximal, sorry, at the papillary region, so fluid becomes from 300 to 1200, right? So in the different region, we have seen outer cortex, suppose for example, inner medullary, outer medullary cortex, inner medullary cortex, and papillary region. So when this fluid reach from the proximal tubules to the outer renal outer cortex to the papillary region, so the fluid becomes hyperosmolar up to 1200 milliosmoles. So now nature has played a trick. It has folded the loop of Henle. If it is straight, what will happen? So see here, the loop of Henle is folded. It is pin, hairpin like structure here. So it increases the osmolarity up to the papillary region. If it was not, it is not bended, then whatever the concentrated fluid is coming, it directly thrown out by the means of urine, right? So you cannot maintain the hyperosmolarity and you lose lot of proteins into the urine, right? So here the nature has played a trick that it has folded this uh, tube, and when this fluid comes here, then in this ascending loop of Henle, thick part, it founds some cells or transporters, and this concentrated fluid, when it comes here, then all the solutes are again reabsorbed into the interstitium. So this region is called as the dilution region or the air diluted portion of loop of Henle or the nephron. So now up to here, it must be clear to you. So this is called as the counter current mechanism. The capability of nephron making hyperosmolarity, this is called as the counter current mechanism. As fluid moves forward to the next part of the loop of Henle, fluid becomes diluted or hypoosmolar. So here in this diagram you can see. So I will try to explain this diagram. So here you can see first this diagram is very important. So here in this black one, you can see from this region to this region. So it is water permeable. And from this region to the late distal convoluted tubule. So this region is water impermeable, right? So first here this region water is permeable and this region is water impermeable, right? So here you can see when the fluid it comes to this region. So here all these rods are it is thrown out by the P cell and this upper part of the distal cannulated tubule, then the fluid becomes hypoosmolar. So how much it becomes hypoosmolar? It nears about the 60 milliosmoles per liter. So when the fluid move forward, so again, uh, so this again fluid moves forward. This hypoosmolar fluid it comes. So here again I will tell you one thing that so from this region to up to the late distal convoluted tubule, this area is impermeable to the urea, and whatever urea is coming from the filter region, it comes to the end or the late distal convoluted tubule, and it does not found any way to reabsorb into the interstitium. So here urea is concentrated. So what is the urea? What is the role of urea? Is coming from the catabolism and other sources. So urea is coming here and not getting reabsorbed. Then the concentration of urea increases and it reaches to the last part of the nephron and the inner medullary area collecting tubules, recycling this part of yellow loop of Henle. So in absence of ADH and what will in absence of uh, ADH. So what is the role of ADH in the uh, maintaining the osmolarity? So here I have shown again one diagram. So this you can see the region here is the cortex region, outer medullary region, inner medullary region, and the last one is the papillary region. So here in the first part, you can see this region, the cortex region. So the osmolarity of fluid is 300, and in the outer medullary region, the osmolarity is 600, and the when fluid moves down up to the inner medullary region, the fluid osmolarity becomes 900, and when it comes to the distal convoluted, sorry, in the papillary region, so fluid becomes 1200, right? So now we will try to understand the counter means opposite way. So here vasa recta plays an important role. So vasa recta is like a dumb thief, right? So when it carries this one, uh, suppose fluid getting filter from this here, right? So the plasma it get filtered here and the fluid comes here, it getting increases, right? So here the fluid is getting filtered and here the gaining here the osmolarity of the vasa recta it increases. Suppose here it is 300, right? So here also 300, 300, then it get increased 600, then 900, and at the last region it is 1200. So it is like a dumb thief. It functions like a dumb thief. Suppose a thief is there and he enters in a house and he has found some valuable things in the first living room. Then he went to bedroom. So when it that thief went deep into deep, he carries some, he found valuable thing. So he's taking more and more that things into his bag. And when he's returning back, so he is losing one of the things again and again. He is losing all the things. And when he get out of the house. So he is having only very small things with him. So vasa recta is also working like that. So when the fluid is going through the region, different region of the vasa recta, so the osmolarity of the fluid is increasing, and when the fluid is going into this direction, so it loses the osmolarity of the vasa recta fluid. And at the last, there is only near about 325 milliosmol per liter in the last region of the vasa recta. So now here in this diagram, you can see, so this is ascending loop of Henle and this is descending loop of Henle. So first it is 300, then 600, 800 and 1200. So after when it is going to the descending region, so it becomes 1200, so it becomes 1800, 600, and at last it becomes 325. So in this region, so vasa recta, you can see cortex, outer medullary, inner medullary, and the collecting distal collecting tubules. So here this works like a, so because the walls are freely permeable to water and solute. So here, so losing H2O, it decreases the hypoosmolarity, and gaining solutes also decreases in this region. It means vasa recta is maintaining the osmolarity of the interstitial fluid. So losing water in this part, gaining solute. So this it means vasa recta is maintaining the osmolarity. So countercurrent mechanism, loop of Henle. So counter vasa recta is considered as counter current exchanger. You only exchange, you cannot keep, it only maintains the osmolarity of the fluid. So counter current multiplier, generation of counter current in the loop of Henle, and vasa recta functions like counter current exchanger. I respond to maintain the, is responsible for the maintaining the osmolarity. So here now we will see the role of ADH, how the ADH works. So now here you have to understand this diagram. So when the fluid reaches to the distal, late distal part of the loop of Henle, so it found some cells in the distal, late distal part of the loop of the Henle. So on these cells, when the ADH it comes from the circulation, so it does not directly enter. It does not directly enter into the cell like the aldosterone. So in the last lecture, we have seen aldosterone is having ability to directly enter into the cell because aldosterone is lipid soluble, but this ADH is a large peptide, so it cannot enter into the cell because it is not lipid soluble. But this ADH is having some receptors on these principal cells, and these receptors are called as the serpentine receptors. So these serpentine receptors is having 7-pass. It is also called as the 7-pass receptors. The chain it folds 7 times. So this ADH binds to this receptor, and this receptor get stimulated, and it when this biological switchers or these receptors get stimulated, it will stimulate the adenyl cyclase chain, and this adenyl cyclase will bind to the next protein. So this protein used to increase the cyclic AMP, and this cyclic AMP also stimulate the protein known as the protein kinase. And what this protein kinase do? This protein kinase, there is one protein that is known as the aquaporin. So this aquaporin is having the aqua means water channels. It is having some channels. So this will switch these aquaporin filaments to the basolateral, to the luminal membrane, and this aquaporin, it makes some water channel. And whatever the water is present is coming from this hypoosmolar fluid. So again, due to these water channels, which is made by the ADH, and it increase or it stimulates this cycle for the formation of the H2O channels. So this suppose 60, it is fluid is hyposmolar. Then this 60, it loses the water, right? So water is reabsorbed and the fluid it is coming to the last part. So it becomes hyperosmolar. So ADH receptor in a 7-pass hormone looks like the receptor. Then ADH receptors on the basolateral side of the membrane. ADH is not a lipid soluble. So if your blood is having the ADH in the good concentration, so ADH through paratubular capillaries. So other source to the principal cells of the nephron P cells are present in the collecting tubules of the late distal tubules. So when P cells are under the influence of ADH, retains H2O. ADH is peptide hormone, so it is large hormone is present on the surface of the cell. So receptor enters if there is no water pores, then ADH, here is the flow chart. ADH bind to the receptor, stimulate the biological switches, then stimulate the adenyl cyclase, increase in cyclic AMP, protein kinase, vesicle loaders move to the liminal side, and this liminal side, it will make reabsorbs the water, and in absence of ADH, you can pass, patient can pass the diluted urine. But in presence of ADH, so the water is reabsorbed and patient can pass small volume and concentrated urine. So high concentration of ADH, it will work on P cell. So P cell highly permeable to H2O, and patient pass the concentrated urine. So whatever the H2 is, it is reabsorbed. And another role of the a good doctors knows the another role of the ADH. So ADH loses more solute osmolality, become hyperosmolar. So here it also helps in stimulating these transporters, that is the sodium, potassium, and 2 chloride. So can become again hypoosmolar, and also ADH also increases the urea transporter, is also increasing medullary interstitium and hyperosmolarity. So role of ADH and the central nervous system. So again, how this ADH is secreted into the blood. So in the interior hypothalamus, there are some cells known as the osmoreceptors. These are very specialized type of the cells called as the osmoreceptors, which are located in the interior part of the hypothalamus. So in hypothalamus, so under the hypothalamus anterior pituitary gland and the posterior pituitary gland is present. So in this diagram, you can see here in the upper diagram, the specialized cells. So when this fluid or the blood, it is going through this area, so this osmoreceptor sense. So when the fluid gets hyperosmolar, so when there is a slightly increase in hyperosmolarity of the blood, the cells osmoreceptors sense which are sensitive to the blood osmolarity. So it will stimulate supraoptic nucleus, and this supraoptic nucleus develop repolarize, and it starts synthesis of ADH, and that ADH is stored in the vesicle, and this vesicle comes reaches to the nerve ending. This nerve ending carrying impulse and releasing of ADH vesicles. This ADH comes to the circulation and finds its receptors on the principal cell. And depolarization of deposition of aquaporin and formation of water channel. Aquaporin make channels H2O in the luminal membrane, and fluid increase H2O reabsorption into the interstitium, and fluid becomes hyperosmolar. So today, I think we will stop here, and in the next lecture, we will see about the GFR and factor affecting the GFR, and we will try to finish it very quickly because your exam is also coming. So tomorrow also I will post one my lecture, and meanwhile you can go through. So we cannot upload the high MB or the high content material into this. So that's why I am going through this Zoom, and we are not able to upload the videos. So that's why we are running with these slides. So these are some notes. You can also follow these notes, and you can make your notes from the books. So thank you, students. So tomorrow we will see about the GFR and the factors of the GFR. Thank you. Thank you so much.